Have you ever wondered what Cas9 chimeras are and the role they play in DNA cleavage? Cue the entrance of Cas9 chimeras, the maestros of the genomic orchestra. They're not just proteins, they're a powerful tool in the world of genome editing. These chimeras or hybrid proteins are created by attaching a protein to Cas9, which is a part of the revolutionary CRISPR system. This fusion enables something quite extraordinary: genomic targeting. Imagine a GPS system that navigates the intricate roadmap of our genes. That's what Cas9 does. The added protein, on the other hand, can modulate or adjust the activity of Cas9. This clever combination allows us to precisely edit genes, turning them on or off, or even modifying their function. Cas9 chimeras are not only significant for DNA cleavage, but they also unlock a myriad of possibilities for scientific exploration and understanding. So in essence, Cas9 chimeras are the maestros of the genomic orchestra, directing the symphony of DNA cleavage. Now that we understand what Cas9 chimeras are, what exactly can they do? Well, in the world of genetic research, they play a significant role in manipulating gene expression. One of these roles is through something called CRISPR interference, or CRISPR for short. This is when a Cas9 protein is fused with a transcriptional effector, like the repressive CRAB domain. This fusion allows the downregulation of gene expression through transcriptional repression. This technique offers a significant advantage over standard RNA interference methods, and it even enables the exploration of non-protein coding genes. But what if we want to do the opposite and upregulate gene expression? Enter CRISPR activation, also known as CRISPR. Here, Cas9 is used to activate gene expression. For instance, when Cas9 is fused with transcriptional activation domains, such as VP64, the result is an increase in the expression of the desired genomic loci. If we want to maximize this activation efficiency, we can use a synergistic activation mediator, or SAM, where the Cas9 fusion contains several activation domains. However, not all Cas9 chimeras have nuclease activity. Some are used in epigenome studies, where Cas9 fusions with chromatin-modifying enzymes come into play. These fusions are utilized to visualize protein DNA binding sites, to examine chromatin structure, or to conduct research on histones post-translational modifications. We've also seen Cas9 chimeras being used in the development of editing nucleases, such as ZFNs and TALENs. In these systems, two discrete nucleases recognize and bind to specific sites at opposite DNA strands. The assembled FOCI dimer then specifically cleaves the target DNA. In the CRISPR-Cas9 system, the DNA site is recognized by base complementarity between the genomic DNA and sgRNA, which is associated with TRACA-RNA, and loaded into Cas9 nucleus, which performs the DNA cleavage. From regulating gene expression to visualizing protein DNA binding sites, it's clear to see that the applications of Cas9 chimeras are vast and crucial. Scene script. Let's recap our discussion on Cas9 chimeras. In the grand orchestra of genetic research, Cas9 chimeras are playing a pivotal role, not just in DNA cleavage, but in a myriad of applications. They are the versatile instruments fine-tuning the symphony of genomic editing and research. We've seen how these chimeras, through their fusion with various proteins, can modulate the activity of Cas9, allowing for precise genomic targeting. This capability is akin to a maestro directing the orchestra, ensuring every note, every genetic sequence is perfectly aligned. We've delved into the concept of CRISPR interference, or CRISPR, where a Cas9 protein fused with a transcriptional effector, such as the repressive CRAB domain, can downregulate gene expression by transcriptional repression. This technique offers a significant advantage over standard RNA interference techniques and opens up new avenues for investigating the function of non-protein coding genes. In contrast, we've also explored the opposite approach, CRISPR activation, or CRISPR. Here, the Cas9 can be used to activate gene expression. For instance, when Cas9 is fused with transcriptional activation domains such as VP64, it leads to increased expression of the desired genomic loci. This is like turning up the volume on certain instruments, amplifying their contribution to the overall symphony. Moreover, we've seen how some Cas9 chimeras do not possess nucleus activity. For example, Cas9 fusions with chromatin-modifying enzymes are used in epigenome studies to visualize protein DNA binding sites, examine chromatin structure, or conduct research on histones post-translational modifications. Thus, Cas9 chimeras are revolutionizing the way we understand, interact with, and manipulate our genetic material. They are the conductors, the composers, and the musicians in the grand symphony of genetic research. Cas9 chimeras, a novel research tool not only for DNA cleavage, but a key player in the symphony of genomic editing and research. It's fascinating, isn't it?